tissue. So it's confusing because a lot of them use the root or the prefix myo. We have a myofiber and a myofilament, perimysium and endomysium, and some of the structures are inside the cell and some of the structures are outside the cell. So you just need to kind of take some time with this to look at the overall organization of it. All right. So here's the large named muscle that's formed by the muscle cells that are arranged in clusters or groups. So if we take one of these clusters or groups, it is identified as a fascicle. All right, so I'm going to start with the smallest external structure, the smallest cell, and then we're going to look inside the cell after we finish looking at these components. So each of these red circles is a cross-section through a skeletal muscle cell. Skeletal muscle cells are long. They are the length of the muscle itself. So if you have a muscle that's 8 inches long, the muscle cell is going to be 8 inches long. So what we have here are various models of types of muscle. At the end of the structure, we'll be looking at smooth muscle and skeletal muscle. But this would be like 3 halves of skeletal muscle. Okay? So they are cylindrical in length, in shape, and in cross-section here, this is actually incorrect because it's showing the nucleus inside the cell, and the nucleus is actually out just under the cell membrane. Okay, but these are long cells filled with actin and myosin. So in the diagram up on the screen, each of those little circles is a skeletal muscle cell. A cluster, so those 15 or 50 muscle cells are organized in clusters or fascicles. So I'm going to kind of help to illustrate this on the board. We have these individual muscle cells. So this would be one muscle cell, also known as a myofiber. Okay? And the nucleus would be, they're multinucleated because they're formed by the fusion of many cells. So when we do see the nucleus, it's right just underneath the plasma membrane. So this entire structure, this cluster of cells, is known as a fascicle. Okay? That's a group of myofibers. And a named muscle is going to have many of these myofibers. So I'm going to draw 3 or 4 fascicles. And then we're going to place, I will place the various connective tissue structures around them. And then we're looking at this in cross-section. All right, so I have 3 fascicles up on the board. And around each individual muscle cell, so here, 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 are collagen fibers of loose, irregular connective tissue. So the loose, irregular connective tissue is supporting the skeletal muscle cells and capillaries, which are the smallest of the blood vessels, which of course are supplying nutrients and picking up waste matter from the muscle cells. So this is identified as endomysium. There's our root for muscle again. And this is loose, irregular connective tissue. Then we have dense, irregular connective tissue wrapping around these clusters, kind of forming the banding that distinguishes one fascicle from another. And it helps the fascicles to function together as a unit.
That's the perimysium.
That would be the perimysium, correct. and that will be dense, irregular connective tissue. All right, then after we have all the fascicles bound together, we have the large individual distinct muscle, and that's surrounded by dense, regular connective tissue, the collagen fibers running the length of the muscle, blending together to form the tendon, which is dense, regular connective tissue. On the outside, wrapped around, running the length, all of this is going to be the epimysium. And this is dense regular. So this, along with some of the dense irregular, blends together to form the tendon and actually attaches it to the bone. So this is all the external structures. Okay? Now let's look back at the fascicle. And we're going to pull out one of these muscle cells. An individual muscle cell, remember, is known as a myofiber. So here would be the fascicle, and then pulling out one of the muscle cells, which is seen down here. Now that's kind of confusing because in the fascicle, these individual muscle cells have little tiny red dots in them as well that look like our original muscle cells. So now we're getting microscopic. We have to use the microscope to see these, and we're looking inside the content of a cell. So we started out with the large named muscle, containing each of these is a fascicle. Then we will look at an individual fascicle. Each of these circular structures is now going to be a muscle cell or myofiber. And now we're going to look at a muscle fiber. So this would be the epimysium around a whole muscle bundle. This would be the perimysium around a muscle fascicle. The endomysium around a single muscle cell. And now we're going to take a single muscle cell, and notice we're inside the cell. All right, so within the muscle cell, here are the peripheral nuclei I was talking about. Within the muscle cell, we have bundles of microfilaments. Remember from our cytoskeleton lecture way back in September, what microfilaments were formed of? Actin was the smallest in the 5 nanometer diameter. What was the largest one? No, microtubules are the very largest, but of the microfilaments.
Myosin.
Myosin. So microfilaments were actin and myosin, and then we had intermediate filaments, and then we had microtubules. Okay? So the myosin and actin are the contractile structures. They don't shorten themselves, but they overlap each other and take up less room. So as they slide past each other, the muscle cell shortens. So now we're inside the muscle cell, seeing these cylindrical structures. These red dots that you see here are actually these cylindrical structures which are formed as bundles of myofilaments. So we're going to start with the cell level now. This was ending with the cell level. So we have the muscle, the fascicle, the myofiber. All right? So may I erase the board here? And then I'll draw a single myofiber as we look at smaller structures. Okay? So you have this picture that's up on the screen on page 8, and then the structures I'm going to talk about on page 10. So The larger picture is on page 8, smaller picture on page 10 with a description of what we're going to cover.
The myosin and actin sheath, you want to call it that, it would not be designated into like a tissue then, right?
Right, correct, because you have to have cells to be the tissue.
Right.
So the tissue would be our skeletal muscle cell. So the structures we're looking at in sequence now, we're going to start with the myofiber. So again, that's one muscle cell. Within that, we're going to see myofibrils. And myofibrils are formed as bundles of myofilaments. And the myofilaments are actin and myosin. So we learned them originally as microfilaments, which they are, but when we see them in a muscle cell, they are called myofilaments. Back to our root word for myo. Okay. So this outer structure here is the plasma membrane of the muscle cell. Every cell in the body is defined by a plasma membrane. We give it a specific term. The other root for muscle is sarco. So myo and sarco both indicate muscle. All right. So instead of just calling it the plasma membrane of the muscle cell, we also identify it as the sarcolemma. So this, when we talk about the physiology, the sarcolemma is capable of generating action potentials. So it's going to have voltage-gated and ligand-gated channels in it. And we'll come back and talk about that physiological function later this week. Because we need to open voltage-gated channels in this structure stores calcium ions, and those calcium ions need to be released to trigger the series of steps involved in muscle shortening. And the channels are voltage gated, so we need to get the action potential of our plasma membrane deep inside the muscle cell. If we just generated an action potential at the muscle membrane, it would dissipate and become weaker as it diffused into the muscle cell. So to keep it a strong current, we have that current travel down the muscle membrane via these channels of muscle membrane that invaginate deep within the muscle cell and wrap around these bundles of myosin and actin. So this is a tube of plasma membrane that also carries extracellular fluid deep into the cell. The membrane itself is also known as the sarcolemma. The plasma membrane tube, the invagination, is known as a transverse tubule. Those of you that have made bread, okay, or any kind of dough, think of the outside of that mound of rising dough as the sarcolemma. And when you push your finger inside it, that would be the transverse tubule. Okay, it's going deep inside the cell. And all of these structures right here, you can see how it wraps entirely around a bundle of actin and myosin. So all of this would be the transverse tubule, called transverse for the T-like arrangement at the surface where it invaginates. And it's going to contain extracellular fluid. So it's carrying those high concentrations of sodium ions deep into the cell as well. So it's really taking the outside of the cell deep inward. It's not inside the cell itself. Okay, it's really still an external environment. Now, it interacts, I'm just going to make this a little bit more parallel with this structure. It interacts with a specialized band of smooth endoplasmic reticulum. That's what this blue structure is. So imagine that our actin and myosin, what we see here in the myofibrils. The actin is thin, so let's say we have a bunch of angel hair spaghetti or linguine, all right, and it's mixed up with large round spaghetti. So that's our actin and our myosin. To hold that in a bundle, we wrap it with bubble wrap. The bubble wrap is the smooth endoplasmic reticulum. Because it's in a muscle, we call it sarcoplasmic reticulum. And that's where the calcium ions are stored. Okay. So that bubble wrap is wrapped around each of the handfuls of spaghetti. There's no actual membrane other than the bubble wrap holding the spaghetti together. Okay. So let me draw that. It has a special relationship on either side of the transverse tubule, where it is much wider and more dilated than any other area. So you can see there's a gap there where it's not quite as reticulated. So this area right here is known as part of the sarcoplasmic reticulum. This entire structure is sarcoplasmic reticulum. Smooth ER, which stores calcium ions. And the dilated region is known as the terminal cisternae. Over here would be the same thing. If we were to draw a cross-section of that, let me do it here. Here would be the terminal cisternae. Here would be the sarcoplasmic reticulum and the dilated region, which is here's the transverse tubule and the terminal cisternae. So you can see it's much more dilated than the other areas of the sarcoplasmic reticulum. Okay? So normal sarcoplasmic reticulum, terminal cisternae, and transverse tubule. Remember, this is filled with extracellular fluid, and as we'll see later this week when we follow the physiology, we're sending action potentials deep down inside the muscle cell for the purpose of opening voltage-gated calcium channels in the sarcoplasmic reticulum. All right? Now think of how this related to what we talked about with nerve. Where were the calcium channels that we opened in the neuron? In the end, in the synaptic bulb. So we sent action potentials down the axon terminal to the synaptic bouton where calcium channels were opened. Okay? Here, we're going to be sending voltage-gated from where our neuron synapses, we're going to be sending action potentials along voltage-gated channels deep down inside the cell. And that current is going to open voltage-gated channels in the sarcoplasmic reticulum. Calcium ions are released, and that triggers the steps required for shortening of the muscle cell. The key thing about the terminal cisternae is that the current that needs to open the voltage-gated channels in the terminal cisternae is lower. It's a lower threshold. Not as strong a current is needed for these voltage-gated channels as for voltage-gated channels located over here. What does that remind you of? Where in the neuron did we have an area of high sensitivity where just as we started to open voltage-gated channels, we had an area that was more sensitive that required the axon hillock. So if we could get a graded potential to the axon hillock, and it was strong enough for the threshold potential of the axon hillock, that would create a current strong enough to open the threshold potentials, reach the threshold potentials of all the other voltage-gated channels. So the terminal cisternae is essentially the axon hillock of the sarcoplasmic reticulum. It doesn't take as strong a current from the transverse tubule to open voltage gated channels here as it might over here. So that's a more sensitive area. Once we open those voltage gated channels, the rest of the voltage gated channels of the sarcoplasmic reticulum will open and release calcium. So that's the special relationship we have between the transverse tubule and the terminal cisternae. Okay, essentially it's the axon-hillock area of the sarcoplasmic reticulum. So again, when we cover the physiology of the muscle cell, we're going to trace what happens when the neurotransmitter is released from the neuron, binds to its ligated channel, creates the action potential, and follow it down inside the neuron. Okay, so we will come back and go over that as a physiological process. So what I've diagrammed here, then, are these structures right here. So here's the sarcoplasmic reticulum. Here would be the transverse tubule, terminal cisternae on each side of it, and the rest of the sarcoplasmic reticulum. Okay? Now that sarcoplasmic reticulum, as I indicated, is wrapped around a bundle of actin and myosin. And that's what I want to look at next. So I'm going to be erasing the board when you're done with those pictures. Yes?
The terminal
Cisternae?
cisternae, is that just, I can keep for that and the sarcoplasmic?
It's a specialized part of the sarcoplasmic reticulum.
Okay, and it's always on the,
It's always on either side of the transverse tubule.
okay. As the muscle goes, as the muscle is long, are there several of them?
Oh yeah, there's hundreds of transverse tubules, and on each side of them would be a terminal cisternae of that sarcoplasmic reticulum. The entire length of the muscle cell. So here we have two transverse tubules with terminal cisternae on each side of them. That would just continue on down the length of the muscle cell.
So there would always be a tubular between them and the plasma, the plasma?
The transverse tubule?
Yeah.
Yeah, I mean this structure here is just going to be repeated over and over and over the entire length of the cell. Okay, so may I erase the board? All right, so before I erase all of the picture, remember that the sarcoplasmic reticulum is wrapped around a bundles of actin and myosin. So extending, as you can see on the left side of that diagram, we're going to have thick myosin fibers. And in between each of them will be thin actin, or around each of them, in a ratio of 6 to 1. And so this is the myofibril. All right? So this is actin. This is myosin. And this entire structure then is the myofibril. So if I was going to be entirely accurate with my picture, we could kind of draw this through, because you'll be seeing it in between. And then you would have actin like that. Okay? So that myofibril is just the bundles of actin and myosin. There's no other membrane around it other than the sarcoplasmic reticulum. The way it's drawn artistically, the artist here put this black band around it. That's not, shouldn't be there. It's just, like I said, if we could hold a spaghetti in a circle like that without anything else around it, that's all we'd be seeing. It's just a bundle of dry spaghetti. All right? There's no other membranous structure around it. Okay, so here are the two proteins, that's fibers, that are involved in this, is actin and myosin. So you have actin first on your lecture notes, and that's what's shown down here at the bottom of the screen. So actin is two things. When we talk about globular actin, individual round protein. So just one of these beads is active. If we take a string of those beads, take 2 strings, and twist them together, that's the filament is active, or a strand of active. So it's a little unfortunate that we use the same word both for the strand as well as for the individual protein. So that's our thinnest filament. Five meters in diameter. So if we take a string of beads, and we take another string of beads, and twist them together, that's the filament is active, okay? So one actin molecule is the globulin form. The entire strand is the filament. Now it is associated with a couple of other proteins that you can see here. In the groove, not quite lying in the groove when the muscle cell is relaxed, is a long protein identified as tropomyosin. Tropomyosin functions to block the binding site on actin for myosin. So when the muscle cell shortens, all right, it shortens because actin and myosin slide over each other and take up less space. So let's say that from my fingertip to my elbow is a myosin filament, all right? From this hand is actin. Notice the distance between my elbows when actin only partially overlaps the myosin. That would be the muscle cell as it's relaxed. When the muscle cell contracts, it does so because myosin reaches up and grabs onto the actin. It can't do so when the tropomyosin is in its location. It's blocking that binding site. And as myosin grabs onto it, it pulls. And let's go and pulls again, let's go and pulls again. And now notice the distance between my elbows is shorter. That's how a muscle cell contracts. Actin doesn't get shorter, myosin doesn't get shorter, but they overlap each other more. To do so, myosin grabs onto actin kind of like a tug of war and kind of pulls it in. It can't grab onto myosin when tropomyosin is in position. Okay? So tropomyosin blocks that binding site. So it blocks the actin binding site for myosin. What keeps it in place, what keeps it from moving aside and exposing that binding site is a small protein known as troponin. So troponin's function is to hold the tropomyosin where it is. If troponin's not there, the tropomyosin actually moves deeper into the groove created by the twisted actin. Right? So let's say that now my two forearms are two actin strands that overlap like this, and the myosin kind of fits. Let me put it in there after I wrap my hands. Okay, so the myosin just kind of fits in there loosely and covers up the binding site. Now push it in deep. When it gets pushed in deeper, now the binding site is visible and available to myosin. Okay, so what keeps it out there is troponin. As soon as troponin comes off, the tropomyosin moves in deep and exposes that binding site. So the function of troponin is to maintain the position of tropomyosin. It keeps tropomyosin from moving. And thus exposing the myosin binding site. Guess what causes troponin to actually leave the actin filament? Calcium ions. So when the sarcoplasmic reticulum releases calcium ions, they bind to troponin. Troponin comes off of the tropomyosin. The tropomyosin moves deeper into that groove. Myosin starts pulling on the actin. Okay? So those are all the sequences of steps we'll go over starting Thursday and Friday. So this is a calcium binding protein. So that's actin. That's the thin filament. Now let's look at myosin, which is the thick filament. A single protein of myosin, rather than being the sphere that the actin is this golf club shaped protein. That's the single globulin form. The filament form is when we bundle hundreds of them together and all these little heads are sticking out. All right? So we have one myosin. This happens to be an enzyme that takes ATP and breaks it down to ADP. It is the site that will bind to the actin. Okay, so that's called the head of the myosin filament, and it's an ATPase, an enzyme that breaks down ATP. So that's one protein of myosin, but when we have the filament, we actually have lots of these with their heads on both ends. And that would be the myosin filament here. Each little golf club-like head will bind to a site on the actin filament. And as we'll see on Thursday or Friday, when it binds to that head, it flips. It's kind of like a, I don't know how many of you use little slingshots or little, my dad got so mad at my sister when she thought she had bought him the best Christmas present ever with her own money. We were living in the Philippines. And she had bought a little wooden gun that shot a pellet by pulling back a rubber band. That created the potential force, and when you pulled the trigger, I think either the rubber band or something flew out. And she had bought it with her own money that she saved up, and all he saw was a gun, and his daughter was playing with the gun, and even though it was really just a fancy slingshot, she still remembers to this day how disappointed she was in his reaction to her special present. But by pulling back that rubber band and locking it, you've stored energy in that rubber band. As soon as you release the cock on the trigger, boom, that energy sets off and shoots the rubber band. All right? So that's what happens with these heads. As we'll see on Thursday and Friday, the energy from the ATP molecule takes the heads and pulls them back and keeps them pulled back like this. As soon as it finds an appropriate site to bind to on the actin, it swings forward like this and shifts the actin towards the center. And then it lets go, requiring ATP, does it again, and just keeps pulling in the actin. So that's how that segment of the muscle cell shortens. Okay? So I want to finish up lecture talking about the structure of the overlap of actin and myosin. It's the specific unit of, the smallest unit of contraction, and it's known as a sarcomere. So back to our myofibril, the actin and myosin are overlapping in specific arrangements. So the red line here is the myosin. The blue lines are actin. And you can see that all the actin filaments are lined, all the myosin filaments are lined up together. Where we have myosin, the muscle cell looks dark. Where we have only actin, the muscle cell looks light. So we look at this view. I'm going to go for it. So this is electron micrograph view here of a part of a sarcomere. So see this, how dark this is right here? That's because that's the width of myosin. The white area is where we have only actin. All right, so myosin, here's actin, and the actin is going to overlap the myosin. If it doesn't overlap, the myosin can't grab onto it and pull it. So that creates a light and dark pattern. If I move this image down here up, it creates this light and dark pattern. Where it's really, really dark is myosin. Where it's light, we have two actins. Okay? And the actins are attached to a band, a disc of protein here. So if we look at a light microscope, if we look at an image of that, those of you in the front may be able to see that this is a band of light and dark. Here we can see it in this diagram. All right? And even in the light microscope, you can see that pattern of light and dark. Here you can see it in a black and white image. So the light areas are actin only. The dark areas are actin and myosin. So those myofibrils are lined up throughout the entire cell so that the myosin are all in a row and the actin are all in a row and so on. So let's look at now a sarcomere. Each myofibril is divided into a series of sarcomeres. And for every myofibril, those sarcomeres are lined up. So we're going to look at, let's see if I can find a better picture here. I'll go back to this one. So I'm going to put down the myosin first. So we're just looking at one myofibril here, a part of one myofibril with myosin heads. And we'll place in the actin. It'll be relaxed, so it's only going to overlap the myosin a little bit. So each actin is inserted into a protein, series of proteins, and this is called the Z-disc. And then we have other actin filaments extending beyond that. And then we have another sarcomere creating more myosin here. So this just goes on and on the length of the muscle cell. Okay, so there's our pattern from this from the end of this actin to the end of that actin is identified as a sarcomere. So from this to here is a sarcomere. That's the shortest unit of contraction in a muscle cell. Remember, the actin and myosin don't get shorter, okay? So the two ends, called the Z-discs or Z-lines, move towards each other as the myosin pulls on the actin. So Z-disc to Z-disc is a sarcomere from here to here. Now where we have where we have myosin is going to be the darkest because it's the thickest filament. Plus it has the overlapping actin. All right? So the end of each myosin, the width of that structure is our dark band. And this is known as the A band. It stands for the word anisotropic. It's not reflecting the light back in the microscope. So the A band is all of this portion that is dark. Now when a sarcomere shortens, is the A band going to become any more narrow? No. Because remember, the myosin doesn't get shorter. Just the ends of the sarcomere are moving closer together as more actin is overlapping the myosin. So the A band itself isn't getting any shorter. That's determined by the length of the myosin. Now, within the A band, we have a lighter region where there is only myosin and the actin isn't overlapping it. So that lighter region is identified as the H band. It's a subset of the A band. Will that change in width if the muscle cell is contracted?
Yes.
Yes, it will become more narrow because the less of the A band is going to be only myosin. So as the myosin pulls on the actin, the H band is going to get very narrow or almost disappear because there's going to be less free space. Okay? So let's say that this ruler is the A band. And I'll just use a colored ruler here to make a difference. So this would be an actin filament overlapping the myosin. The A band is the width of the myosin is the width of the A band. Here's the actin overlapping it. And if I have another actin filament over here, the distance between the two yellow rulers is going to be the width of my H band. Does that make sense? Okay, this is actin, and as it overlaps the myosin, the free space, the distance between the ends of the two rulers here, here and here, is going to be the H band. Now as more of the actin overlaps the myosin, notice what happens to the width of the H band. It becomes narrow as the muscle cell contracts. When the muscle cell relaxes, the H band becomes wider. Okay, so the A band doesn't change, but the H band can. Now there is a series of proteins that actually connect the myosin structures together, and that creates the M line. Now, we have one other structure here. In the other sarcomere, with the other actin filaments, we have this area that is light. Okay, it's lightly colored because it's only actin, which are thin filaments. So this is known as the I band. It actually takes part of 2 sarcomeres. But it's light because it has only actin. It will always have only actin. Okay, so let me turn the lights down and get a little bit more contrast here.
Are you gonna go over the proteins of the M line later on, or?
No, that's all you have to know about it. So here is a sarcomere starting from Z disc to Z disc.